And should everyone take themselves off the picture to give more room for the slides, etc.?
Oh, that'll be right. But if people on, yeah, you're right. Yeah, if people turn their video off and and mute, that'll be excellent. Okay, well, thank you, Brian, for giving that acknowledgement to country. Tonight we're and welcome to everyone who's here. Thanks for attending. At the moment, we have 15 participants, including Ross. And tonight we're honoured to have my dear friend Ross Jeffree, who is very condescending, but is actually written up, as you probably saw, as the Chief Scientist, Jeffree Conservation and Research, probably known as Professor Ross Anthony Jeffree AM, who achieved his AM for significant service to conservation and the environment and to nuclear science and to education. Ross, in his working life, has worked for ANSTO and University of Technology, and he's always approachable. And he's also pioneered some of this DNA barcoding for insects in association with SSEC, Southern Shire Environment Centre, in the national park. And he's now giving presentations to tonight MPA, and Thursday will be Friends of Royal. And he's tonight talking about the unique terrestrial invertebrate biodiversity and how their DNA barcodes are being added to a global database called the International Barcode of Life. And he'll also tell us, I'm sure, of the sheer numbers that have been discovered new species. So I won't say any more. Ross, over to you. Thanks for being
Okay,
here.
thanks, mate. Look, I just mentioned, Jeffrey Conservation Research. It was a huge group of people, but then the dog died, and so there's only me left now. So that's I use that as a sort of a name for what I do multifariously. So yeah. Okay, so I'll bring my and I'll just share. Oh, hang on. Can you see that?
No, not yet. Share screen.
Oh, where the hell is that? Hang on.
Um,
I should go to share first.
Down the bottom. Yeah, go down the bottom.
Down the bottom to share.
Just a bit.
Oh, okay. Yeah, thank you. Thank you.
You're underway. It'll
And
come up
then
in a moment.
slideshow
Yeah, you got it. Slideshow from the beginning.
from the beginning. Okay, thanks so much. Yeah.
Let's go.
Okay. Oh, hang on. Yeah, so I just want to say that there's three groups making this project happening. I mean, I really personally think it's a very exciting project, and we're lucky to be able to do it. So we've got the volunteers from the Southern Shire Environment Centre, and we are, we're supporting the National Parks and Wildlife Service Eco Health Scorecard that's basically focused on the mammals and the birds and the other vertebrates. And we feel like we're complementing that by looking at where the real biodiversity is, which is in the insects, as I'll show you. But we're also supporting the International Barcode of Life, which is a very exciting project of I regard as of planetary significance. And so it's such a privilege to be involved in this project. And also it's telling us so much about what is in the Royal National Park, to the point where I think if you add what we are finding from the Eco Health Scorecard with what we're finding out about the insects, it must be perhaps one of the best characterised national parks in Australia with respect to terrestrial biodiversity. I don't say I know a lot about insects, but I'm finding out more, and I'm finding them quite interesting and actually inspiring. And I think here's an example of how they may well have inspired someone else as well. There's an uncanny similarity between the Australian termite mound or castle and La Sagrada Familia that was designed by Gaudi. So either they were both they both received divine inspiration, or Gaudi got a look at what Australian termites have achieved and design something around that, because we know he was very much inspired by organic form. So that's just something of an interesting curiosity, I think, but certainly insect and inspiring. So right up front, I want to say something about the people that are making this project go in terms of the volunteers. So Brie McClafferty, she's our Eco Health Scorecard liaison person within NPWS, and she goes out and collects with us when required. Melissa Harvey is a geneticist, Sue McTaggart, big in wires, and myself, So we represent the volunteers from the Southern Shire Environment Centre. But the real heavy lifting with respect to the science is Paul Hebert and Kate Peraz from the International Barcode of Life that are at the University of Guelph, and I'll say more about Paul soon. Anthony Chariton and Jono Shaw from Macquarie University, they're linked into this EcoHealth scorecard stuff and DNA, and they helped us a lot with setting up nets and providing some of the 95% alcohol that we required, and quite a bit of it as we went along. So I just want to acknowledge here Professor Paul Hebert. He really is the father of the DNA barcode. He invented it, and he's quite a character. We go back a long way. I was doing honours in the zoology department. He'd just come from Cambridge University with a red-hot PhD. He was a bit of a whiz kid, but he really liked to get out in the field. We used to go down to Boiler Creek looking for powerful owls. He helped me quite a bit with my honours project, etc. So I never forgot that. He really loves Australia. He works very hard, 7 days a week, but at the end of the year, he brings his family back to New South Wales to get a bit of sunlight and longer day lengths, and has had this long-term connection with Australia. So his mission in life, until biology retires him, is to use this DNA barcode approach for the discovery and the identification of all multicellular life on Earth. That's all, okay? So it's a wonderful mission, and for his long-term efforts and research at the highest level, he last year received this Nobel Prize in sustainability. Which is amazing. What it means for us really is all the data that we are getting out of the Royal National Park is at the highest level of science that you can possibly get in the world. So, and basically for free. So it's a remarkable achievement to be able to tap into this work that Paul is doing. Now, let's talk about the ecological necessity of insects. 70 to 80% of all biodiversity, and that includes marine, is insects. And the ecosystem services they provide, pollination, pest suppression, decomposition, nutrient recycling, they really are ecosystem engineers. They're not as charismatic as a bilby or a brush-tailed bettong, but yet they're out there doing the job. And of course, they provide this stable source of food for other species. At the same time, it's now being realised we're in the middle of an insect crisis. The numbers are declining, and it's probably the ecological impact that you hear least about, but the people in the know say it's just as significant as climate change impacts on biodiversity at this point in time. Here's a few biodiversity realities. Most biodiversity is terrestrial. 70 to 90% of terrestrial biodiversity is insects, and most insect biodiversity has wings, okay? And this is exactly what we are sampling with our protocols. Now, this is a paper, it's so pivotal, this paper, in determining this decline in insect biomass over the last 27 years. It was a study that was undertaken at 63 protected sites in Germany using these nets, which we are using as well, but in protected areas. So there's no degraded habitat, but they have seen this remarkable decline of biodiversity over this time, and I'll show you some of the data. Now, note that the scale on the y-axis is a geometric scale. So arithmetically, it'll look much more dramatic, but you can see this steady decline in biomass over that period. If you look at the annual biomass, you've got a hill because it's in the northern hemisphere, but you can see this decline as you move down to the orange dots. And here is just a sort of an average overall showing this systematic decline with time. So that put a lot of people, put them on their back feet with respect to the severity of this problem. At the same time, I want to mention the value of insects, and this is a very good example of how valuable insects are to us. This tiny little midge pollinates the cacao flower. If it wasn't for this midge, we wouldn't have any chocolate, and Johnny Depp and Juliette Binoche wouldn't have been able to make that lovely film, which many of us have probably enjoyed. So I don't know if Johnny Depp realizes how much of his career success is due to this little midge, but there you go. I've got a bit of a love-hate relationship with midges. I did work at ANSTO when it was AAEC. My first gig there was to look at this purple striped gudgeon and look at the diet, see if the diet actually reflected the degrees of pollution in the Finniss River that was receiving contaminants from the Rum Jungle mine site. So I looked at 737 fish diets and found a lot of ceratopogonid midges and other things in there. And yeah, I haven't forgot that experience. I felt like I was paying my dues to be a biologist by looking at so many fish guts. Let's have a look at another decline, which is attributed to this the demise of insect biomass. Bird populations declined in the US by 29% since 1970, particularly the insectivores. Here's what the decline looks like. I mean, these numbers are terrifying, really. If you look at these data in more detail, you can look at the changes since 1970, coastal, Southwest arid land, South America. You can look at the degrees of decline over that period of time, which is being attributed to the decline in insect, as well as habitat destruction, agricultural intensification, etc. Now, let's talk about the barcodes themselves. Here's a simple schematic that is what it's all about. So for an animal like an insect, you might take a small portion of one leg of a preserved specimen, do the DNA analysis on it, and from that, one part of the DNA, construct the barcode. And these barcodes identify the species. So once you've determined the barcode is forever. The taxonomic name of the insect may change according to what taxonomists deliberate upon, but the DNA barcode never changes. It's solid. It's a way of concisely and unequivocally determining which species you have. Okay, and these DNA barcodes go into a reference library, and then you can get a very accurate determination of the species you're actually working with. So what's actually involved in doing a specimen? This is about $3. It's incredible how inexpensive this is per sample, and the cost is coming down with time as the technology gets more sophisticated and the throughput is greater. So when we send our samples to the University of Guelph, each individual is databased, it's curated, it's photographed, a tissue sample is taken, the DNA is extracted, the barcode region is exaggerated using polymerase chain reaction, and then it's sequenced, and from that sequence, the data is validated and then uploaded to the barcode of life database, so-called BOLD. So mitochondrial DNA is used for that purpose. And the mitochondria are the little powerhouses that we find in the cells. Between generations, the mitochondria are transferred only through the maternal line. So you don't get any genetic shuffling that you will get with sexual reproduction and nuclear material being shuffled and recombined. So within each individual species, the mitochondria is quite conservative within the individuals of the species, but it's quite different between species. So it's a very good marker for each individual species, and the region within the DNA of the mitochondria that's used is called the cytochrome C oxidase, or COX region of the DNA. So what's a DNA barcode look like? I put one in here for the eastern yellowjacket wasp. This is a typical information sheet that I imagine we're going to get for the 10 or 15 million species of organisms that are out there. You've got a taxonomic diagram, you've got a photo, and here's the DNA barcode. So the barcode is much bigger than you get on your packet of chips, for example. It's got a color variable in there as well. So it's huge, lots of variables, but of course it's got to be big enough to be able to incorporate the 12 to 15 million species that Paul Hebert and his team are thinking we're progressing towards. In terms of the bigger project for the decade, its mission is to address two shortfalls in information with respect to biodiversity, the so-called Linnaean shortfall, which is the fact that we've probably only got names on 25% of species. And the other one is the Wallacean shortfall, which is we don't know much about the geographical distributions, and that's named after Alfred Russel Wallace, who was the father of zoogeography. He did a lot of work in areas to the north of us, for example. And the ecoregion that we are working on, we are sampling from, according to the IUCN, is called the Eastern Australian temperate and subtropical forest region. Okay, so the idea is to do 10 million specimens. I guess from our project with the Royal, excuse me, we will be sending 150,000 to 200,000 individual specimens to Paul. Now, the other bit that's coming up now is the species interaction. We haven't done very much with that, but I'll just briefly mention that as a potential as we go along. So I imagine you're wondering what's the use of the DNA barcodes. Okay, we know that Australia's barcode reference library is very incomplete. And so this is a real impediment with the application of inexpensive DNA-based approaches like eDNA and metabarcoding. And I know within NPA, some of this work has been done in the marine area. So this project is really addressing the big gap that we have in our Australian collections. And the sampling methodology is the so-called Malaise trap. Now, the René Edmund Malaise, a Swedish entomologist, explorer, art collector, was in Rangoon, and he designed in the 1930s, and he designed this Malaise trap, deployed them, caught about 100,000 specimens, sent them back to the Swedish Natural History Museum and worked on those. But it was found that the methodology was so good that many other people started to employ it. It's really good for flying insects, which, as I mentioned, are the major component of insect biodiversity. Here we see some of the regular suspects that helped us with the first setting of the first Malaise trap down at Little Marley. And as we went along, we set 4 traps, one at Little Marley, one at Marley in the heath. It's a bit windy down there, so the weekly sampling over the annual cycle was good, but sometimes we had to resurrect. The net had been blown over or whatever, so the heath was a bit of a problem. We had to actually move the net once because it got blown over and destroyed once. So yeah, that wasn't so much challenging, but a weekly assessment of how the net was setting was important as well. And we set one at the southern end of Lady Carrington Dry in the wet sclerophyll grassy habitat, and one at the ranger station, which was dry sclerophyll. And as I said, sampled every week over the annual cycle. So from the first batch that we sent to Paul Hebert and his team, there were about 7,000 specimens. This is how the data actually came back to us. So every collected specimen is identified taxonomically down to family level, for example. DNA is sampled and then it's archived away. So these samples continue to be owned by us. They're just put in this enormous archive. And that's, of course, it's a very labor-intensive activity for the taxonomists. Paul Hebert and his team, there's 120 of them. About half of the taxonomists, etc. The other half are the DNA people. So it's a huge number of people that are working on this project. And, the amounts of money that Paul has pulled in, it's about $250 million so far. It's a remarkable amount of money, but of course, doing the DNA barcoding of every species on Earth is a big challenge. This is how we received the data for those 7,000 specimens. This is page 24 of 24 pages of a phylogenetic tree that tells us the number and the order or the family of each individual, where it goes to in the BOLD, the Barcode of Life database, and what's the relationships between each of those individuals. So you can see we get an enormous amount of data back. Here's a bit of a close-up. You can see Hemiptera here. These are the bugs. Hymenoptera, these are the wasps or the bees, mainly. Araneae, the spiders. Arachnids could be mites or yeah, it could be probably mites. And then we've got a Neuroptera, which is a netted wing individual, and then a few Coleoptera. So that's just one, a little bit of one of the pages. In terms of looking a bit more at the data itself, out of 7,000 specimens, we had 991 species that were identified. And if you plot the number of individuals versus the species diversity, you can see that the numbers are increasing, and they're not asymptotic as yet. In other words, there's a lot of other species to be determined at each of those sites. If you look at a Venn diagram, you can see there is some overlap between the two and shared species, but basically most of the species are unique to those habitats that we had sampled, even though they're both in the heath. So similar habitat, but geographically they can be relatively unique, and they're only 5K apart from each other. And at that point, the people in Guelph said, look, many species are likely undescribed at that point in time. So this is the next set of results received. And here we've got 65,000 specimens that have been worked with. And what is it? Hang on. Just get this out of the way. And about 5,352 species have had their DNA determined and introduced into the Barcode of Life database. Again, this plot of number of individuals versus diversity shows that, sure, it's curving a bit, but it's got a long way to go, heading up towards 10,000 at that point in time. If we look at the individual sites, we can see that the ranger station, the dry sclerophyll, looks like it's got more species than the two heaths. And Lady Carrington Drive's got a few there as well, but they're all trending up positively and are not asymptotic at that point in time. Okay, if we look again at the way they were analysed previously, we can see there's quite an overlap in terms of the species among the sites, but still there is unique numbers associated with each of the sites. And it's interesting, if you look at Little Marley and Marley, both in heath, but a lot of species they do not have in common. So in other words, that suggests to me if you put a few more nets out in heath, you're going to find even more species. That's not something that we're doing at the moment. I'll mention what we're doing this year, but there's a lot of species out there. If you look at this one here, again, you can see there's overlap between the three habitat types, but a lot of uniqueness in terms of the species composition with the habitat type. Okay, so the extrapolation that has been done in Guelph suggests that at the 4 sites, based on the current data, there's about 10,000 or more arthropod species. And the interesting thing is that 57 to 60% are new to science. They've never been seen before. And this is a very exciting result for us, I think, showing the value, the biodiversity value of our park, but also to the international community in terms of what we've got down there. So now I want to talk about some of the lifestyles of the animals that we have caught. A lot of the insect lifestyles, we might regard them as unsavory. Charles Darwin's had something interesting to say about this a long time ago, and I'll read this out. If men were reared as hive bees, our unmarried females would, like worker bees, think it a sacred duty to kill their brothers, and mothers would strive to kill their fertile daughters, and no one would think of interfering. In other words, bee morality is looked at through a different lens compared to social primates like us, our morality, and it's all about survival and reproduction anyway, and they've been successful for a couple of 100 million years. So yeah, so it's better to not be so judgmental, I suppose, of species that have been so successful, and we've only been around for 250, 300,000 years, and not everything we do is ethical as well. Let's have a look at the composition of the catches where the biodiversity really is. So it's in the flies, and these are some of the representatives. Coffin flies, well known for their ability to dig down 6 feet and drop their eggs into cadavers. Midges, well, we know they're annoying, blood-sucking things. And here's a fly here, and we know they're good at getting rid of decaying matter with their maggots, chewing things up and disposing of them very rapidly. Crane flies, often people don't like them because they look like big mosquitoes, but they're quite an innocent vegetarian. Here's a guy that's quite interesting in terms of lifestyle, the jackal fly. He jumps on the back of spiders and just sits there feeding on their body fluids. Yeah. Now, this is where the really interesting stuff happens. With the wasps, the bees, and the ants, there's about 5,000 species that have been found already. It's incredible in terms of this group being sort of so biodiverse. And, Paul Hebert and his team are amazed by this to the point where they want to write a scientific paper and put the volunteer's name on this because it is such an unusual outcome in terms of their global knowledge about where biodiversity is. So the wasps, the bees, and the ants. And as I've learned, bees are just a type of wasp anyway. We don't get a lot of ants, and so it's mainly wasps. But here's some of the interesting guys, like the fairy wasp. It's probably the smallest flying insect. And for it, paddling through air is like us pushing our way through honey. Its wings are made into little paddles at which it clicks back and forwards at 1,000 times per second. Quite remarkable. And it's quite a parasite as well. These braconid wasps similarly are parasites on caterpillars. And their aim in life is to get an egg into a caterpillar. Of course, they're used in biological control quite a bit. And here's Darwin's wasp, an ichneummonid. It's very good at depositing its eggs into the larvae of other insects. There's more species of Darwin's wasps in the world than there are all the mammals and the birds put together, and probably only 25% of those Darwin wasps are actually taxonomically described. So the biodiversity is quite remarkable. There's also a lot of moths. They're very important pollinators as well. And I just put these two here in. I mean, this one and this one. They look pretty nondescript, pretty comparable, but in fact, genetically, they're in different families. So they're as different to each other genetically as a blue-tongued lizard and a goanna is in terms of their divergence in evolutionary time. Now, I just want to show you some better critter pics. Most of the animals we have caught are quite small, and you can't really appreciate their interesting anatomy and the complexities of their bodies unless you look at some good photos of them. So here's a nice fly, lovely maroon compound eyes, iridescence of the wings. What's not to like about this character? Yeah, he's beautiful. Here's a weevil. I found out weevils have these long noses to drill holes in vegetation so that they can deposit their eggs. But yeah, look at the complexity of the anatomy. It's really something that you don't appreciate when they're just a few millimeters in length, and you just can't see the beauty of them. Here's a netted wing, a neuropteran. You might know the larval form as an antlion, for example, but this is a lovely photo of the wings and the rest of the body there. And here's our ichneummonid wasp with that very long ovipositor that it uses very effectively to deposit its eggs in other insect larvae. Now, I want to mention something else that's happened in this project. Because we're sampling so many individuals, we've got good people there who know what they're talking about taxonomically. Things are turning up that are unexpected. And this is one example of a few. So this is a family described in 2021, Neurocilionidae. The taxonomists are excited. Paul Hebert farms out these unusual finds to people who are specialists in the field. And there's a research group in, I think, Kentucky, who are only focused on the wasps. And so they really know everything about wasps, and they're saying, wow, this is quite exciting in terms of what's coming out of the collections. I know some people are interested in bees, and there's concern with what's happening with honeybees in the Royal National Park. We've found 14 native bees as well as honeybees. It doesn't seem from the numbers we've collected using our sampling protocol that they're taking over from the native bees. And so I know that is a hypothesis that is out there at the moment, and then it's confounded by varroa mites, etc. But anyway, I just wanted to show you that what we have found with the bees. Now, also talking about species interactions, some of the groups that we have caught, the horseflies, the banners, the midges, the mosquitoes, they've probably got mammalian or bird or reptile or even amphibian blood in their digestive system. So that's preserved away, and it's possible to extract some of that and see exactly what they're feeding on. There might be species of vertebrates that we haven't been able to find in our criticams and soundscapes. They might turn up in the DNA in these bloodsuckers. That's something that has a potential for the future. So here I want to, this is for the National Parks Association, I want to pose a question. Should national parks be even further valued as repositories of insect biodiversity that restores ecosystem services to biodiversity, degraded agricultural landscapes adjacent and beyond? I say beyond because of the migratory possibilities, potentials of insects. And just something to support that hypothesis. This is an interesting study that came out of Rajasthan, okay, just recently, 2025. Forest expansion actually increases agricultural output, the new study shows, by 24% in 7 to 14 years after forest expansion. The authors attributed it to increased pollination activity, as well as a little bit of increase in rainfall. I thought there was such an important study that I passed it on to WWF forestry program as well. Yeah, I think it's one of these pivotal studies that could start to change the paradigm with respect to the value of forests to agricultural lands. Now, I want to talk about the dollars. Economics is important in these projects, just to show what we're getting for free, basically. So the cost recovery value of the annual sampling project at the 4 sites, the taxonomy, DNA barcode, it's $360,000 so far. We've got more samples to send. There's enough input coming into the eyeball for enough for several other national parks in New South Wales to be sampled. Bria McClafferty, our liaison officer within NPWS, organised a meeting between Paul Hebert in Canada and people working in 6 to 8 of the other national parks in New South Wales to explain what is possible, and many of them were very positive. And I believe from Brie that the sampling program is about to begin with a couple of nets in the greater Blue Mountains National Park. So what are we doing ourselves within the SSEC? We've got 3 new sampling sites in the Royal at different habitat types. So Lady Carrington Rainforest open and closed. So we're trying to optimize the number of insects that you might have up in the canopy that you don't get on the ground, but apparently open areas are more likely to attract them down, and we're doing a closed one as well. And then at Kelly's Fall, we've got a wet sclerophyll bushy, which we hadn't covered before as well. So we're sampling, these ones are being sampled every 2 weeks, because the work on the DNA indicates that it still has its integrity even after 2 weeks of sampling. So yeah, that's going to continue the rest of this year. I also went to the NPA Marine Parks Forum. I had a chat to some people about the possibility of doing DNA barcoding in marine national parks, existing or notional. Prior to that, I got in contact with Paul Hibb to see if he was enthusiastic, which he was. So there's a possibility there. It's a matter of talking to the right people now about whether there would be interest in thinking about the protocols for this sampling. But it could add a lot of value to the argument for biodiversity conservation and where you put a national park in terms of optimizing protection of biodiversity, which I guess we really don't know at the moment. We know about different habitat types, but we don't know about biodiversity. So that's something that has been initiated, but we'll see where it goes. There's a lot of other things happening with eyeball. South Africa, all the national parks are being sampled. France is announcing a sampling program soon. So Kruger National Park has been sampled already on this annual cycle, weekly samples. 25 nets have been deployed in Kruger, and apparently the biodiversity of insects in Kruger is only matched by the Royal National Park, and we've only had 4 nets out. So that is really quite intriguing at this point in time, and the rest of the national parks are being done as we speak. So this is the last slide. the vision is barcoding terrestrial invertebrates in every Australian national park. The investment is small. It's a matter of having 95% ethanol available to you, and it's a matter of having a heat sealer for the plastic bags. So yeah, so that's how we've proceeded. So incredible value for money. Okay, cheers. Thanks very much.
Thank you, Ross. Wonderful, consummate presentation. I'll open it to questions or comments.
Yeah, can I ask one?
Yeah, Gary.
Yeah, Ross. Sort of thinking about barcoding in marine environments. What are the advantages and disadvantages of this this form of technology with the eDNA that you're familiar with? That there's been some work on in the local area.
Yeah, look, I think the major difference is that you don't have a comprehensive reference library against which to determine from your eDNA what the species are. That's the impression I've got at the moment. There's a shortfall there. So you don't get as much resolution in your taxonomic identification. You might get to family or genus, but you might not get to species because the barcode isn't there for the species. So I suspect that's the situation at the moment. Yeah.
Okay, we'll go to Brian, who's got his hand up.
And put a question in the chat, Ross, if you want to see it. To begin with a sentence, but
okay.
There's a whole pile of areas that used to be set aside in the European market, in inverted commas. They were called set-asides.
Right.
Where they used to have strips of native plants or vegetation around the farmlands. Just curious if ever that has been measured as to whether it's had any economic impact on the productivity of the farms because of the increased amount of pollination to begin with, but all the other values that comes with it.
Yeah, look, I know something about that. I know that after this decline in biodiversity of insects that was measured in Germany, the German government was so concerned about it that they first of all they banned the neonicotinoids. You know, that's a very lethal group of insecticides, and also they said all this agricultural intensification, which just means removing all the brush and the weeds and etc., that has to stop, and we've got to start putting more. You know, we've got to allow more roughage and weeds and things to re-establish themselves to support the insect populations as well. So and that was Germany, and so I don't know if it spread through the European Union as well, but certainly that was the response from these terrifying results that came out of Germany. But see, the interesting thing in what's happened in Germany is we've seen declines in the protected areas, and so you know it's a little bit intriguing, but their determination was it's probably from these powerful insecticides that are wafting from the agricultural lands, and so that's all I've got at this point in time on that.
Yeah, their protected areas are, of course, a hell of a lot smaller than what we or what the Americans or what South Africa.
I guess they are. Yeah. I guess they are too. So there'd be more impact.
So they would have an edge impact. Yeah.
Yeah. Exactly. Yeah. So it's an interesting area, you know, this getting the mix right between habitats that preserve biodiversity that can do all your pollination and what effect that has on agricultural productivities. But, you know, that Rajasthan study, I think, is a real paradigm shifter. Yeah.
The second paper I sent you the other day was on wildlife crossing in California.
Right.
And it isn't completed yet. But if you get around to reading it, it thrilled me to note that the insects have already taken control of it.
Have they really? Yeah, look, I haven't read that yet. I'll have to have a look at that. Well, that's see, the thing about insects is that they have such capacity for rapidly increasing in numbers. You know, these short life cycles and the percentage of energy that they put towards reproduction means that they can increase exponentially in a very short period of time. And their migratory capacities are absolutely amazing. We think that birds are good at migrating. Well, you know, dragonflies can go several times around the world. That's quite amazing. And I didn't realize it until I started reading a little bit about their capabilities. And I thought, God, it's remiss of me. I've never spent enough time investigating insects, because now I realize how amazing they are. Yeah.
Speaking of, and if Ian, I'm allowed one more and I'll shut up after this, that you were finding a lot of stuff in your traps, but missing things like a lot of the big flying butterflies, moths and dragonflies. Now, I'm assuming that is part of the design of the trap that doesn't pick up the big flying things.
We do get some big flying things in there, assassin, or what do you call them? We do get some larger insects in there as well. I'd say in terms of numbers, it's mainly the small gnats and things like that fly in a swarm and all that sort of thing. I don't think we've got any dragonflies, and I don't think butterflies, but you get a few moths in there as well. But yeah, it's a little bit selective, but it's probably the most comprehensive methodology for getting an idea of insect biodiversity. If you put pit traps in, you're going to get more terrestrial beetles and that sort of thing. And if you put yellow pan traps, you're going to get more bees. And if you go out at night and you put up a screen with a light behind it, you're going to get more moths. And so, they're all a little bit sort of specific to the sorts of insects you want, but as a sort of a methodology that can be deployed and is going to get most of the biodiversity, it seems to be the protocol that people mostly use. Yeah.
Gary's got a question.
Yeah, I'm just wondering about the logistics of volunteers versus more professional people, say in the Royal project. Like what sort of numbers of volunteers and things are involved here?
Well, there were four of us. There were four of us. There were four nets, and there were four of us. And so it was just a matter of dedicating yourself to going each week to twist one bottle off and put another one on, and then take the sample back and basically stick it in the fridge. So it wasn't difficult, but you had to have people who really wanted to do it every week. And
so you guys didn't try species sorting. That was done by the taxonomist, was it?
No, we did absolutely nothing like that. That wasn't our role. Our role was just dedicated to getting the weekly samples and processing them so that the integrity of the DNA was preserved. And then it was pretty easy to send them over to the University of Guelph. It was all paid for. And once you've got them preserved in 95% alcohol, the DNA is going to last a long time. And so it was pretty straightforward. We didn't have to do anything down a microscope or anything like that. So, working your life around weekly sampling, we had to help each other out. If someone was going, well, I'll go and sample your net for a while and whatever. So it was a good team effort, in terms of making sure that we did get the samples done. But all the heavy lifting was really done in the University of Guelph. And that's the great thing about the project. All the quality of the science coming out is quite remarkable in relation to the effort that we put in there. But it wouldn't happen unless we put the effort in. And all those nets were supplied for us. So it's a great thing to be a part of, I think. Yeah, everybody that was involved is really excited about what's coming out of it, and now I suppose it's a matter of sort of thinking of the value of it. The value of the biodiversity in terms of persuading people in policy that we've got something quite remarkable. And but why is it remarkable, and how is it valuable for society and all the rest? So that's why I threw in that Rajasthan study. I thought that was really very pivotal. Yeah,
it seems to me, why go to Mars and hope that you might find a bacterial type thing amongst the dust when you've got all this stuff here?
In the Royal National Park.
Yeah, that's right. That's amazing.
And it was completely unexpected,
No, that's
we'd
right.
have so much
I'm
biodiversity.
amazed. I mean, I'm an entomologist theoretically, but
that's right.
I'm absolutely amazed by the scale and the implications of what's being done.
Incredible. It's exciting stuff. It's like a discovery project,
Yeah, yeah.
Yeah.
Good.
Now I'll stop going on waxing lyrical. Yes, it's quite exciting.
Yeah.
Ross, I might ask a question or two.
Sure, mate.
Uh, I was interested, first of all, in that graph that you had, that illustration of the DNA where you had all the colors.
Yeah, yeah,
Normally, we've just got four
Yeah.
sub bases, and you'd think, red, green, yellow, blue, or something
Yeah.
like that. But you had a whole melange of colors there.
yeah.
What was that
Uh,
indicating, I guess?
um, I just think it's offering more combinations
Okay.
of the DNA in the mitochondrial DNA for fitting within this barcode of life database.
Right.
So I think that's all to me, that's apparently all it represents. It gives you yet another dimension, um, um, which will multiply the numbers that the barcode can hold. So I don't think it represents the different bases and all that sort of
Oh,
stuff.
yeah,
It's
okay.
just
Yeah.
a mathematical way of
Right.
enhancing the capacity of the barcode to include so many species. Yeah, that's the way I
And
interpret it. I could be wrong, but
no,
I,
that's
as I
fine.
said, it's not my field really. But
yeah,
yeah,
And
I'm going to guess that the different colours might represent some particular sequences of A, T, G, and C.
I don't think
Um,
so. But I can, if you're interested, I can find out more about
yeah. Mm.
it. Yeah,
A
but I don't
second que
think. Yeah. Anyway.
a second question is, I know how you can get DNA from cells and things, but how do you specifically focus in on the mitochondrial DNA when you've got a
Well,
whole goo there? Is it fairly?
oh, look, the technology of extraction of the mitochondrial DNA, I do not know. If we had Melissa here, who has a PhD in genetics,
Uh,
she'd be able to tell you
uh,
how they focus on the mitochondrial DNA. But I do not know. It's not
mm.
my field at all.
Yeah.
So I should have said that right at the moment. I didn't know much about genetics or insect,
Yeah.
but
And
I'm the
wasn't
messenger.
and weren't mitochondria considered to be a form of bacteria that had invaded
Um,
the
Yeah, in terms
that's
of
a hypothesis that
um,
been around. Yeah,
yeah.
yeah, yeah.
Yeah,
I think,
that's the interpretation of why they are
Yeah.
in the cell,
Yeah.
as well as a lot of other organelles that you find in a
Yeah.
cell.
yeah,
That it's a mosaic of other species
Yeah.
that have found ways to live together
I
to mutual
contain
advantage.
multitudes.
Yeah,
Sorry,
sort of.
I
like
contain multitudes.
we're a
Yeah.
mosaic of a whole lot of species as well, So and cells are as well. So the mitochondria is apparently a bacterium that was captured by this cell and found a happy home there.
Well,
But,
sorry, you're dealing with prokaryotes, aren't you? Going into forming in part of a eukaryotic organism.
yeah, that's right.
exactly, yeah, yeah.
Mm,
But the thing, as I said, the thing about the mitochondrial DNA is quite conservative
mm,
within a species, and so you don't get this variation,
mm.
which would
Yeah, yeah.
confound you from discerning between species. And so that's why it was zeroed in on. Yeah, for a very accurate differentiation between species.
And the
Yeah,
third question is, Ross, if I may, we've all, I guess, learnt 101 biology that the concept of a species was initially a species level arises when two different organisms can't reproduce sexually. That's the difference. So if
yeah,
so you've got that reproductive hurdle
isolation. Yeah, yeah,
to distinguish between species. I
yeah,
do
sure.
note that in the floral world and the world of plants, the definition becomes a whole lot looser than that, or they use other definitions to find species. What I'm sort of going
Well,
with this is, what's your criteria for species when you're actually looking at these insects, given that they're all reproducing sexually and there is differences, at which point do they sort of differentiate this is species one, not with, you're saying you've found 900 species or something.
yeah.
What's the criteria?
Well, I think the fact that um, within a group of individuals that are only varying in their mitochondrial DNA by 1 or 2%.
Okay.
And you've got another group that vary again within themselves by 1 or 2%, but they vary between each other by 5 or 6%. That then you've got two species. The interesting tie-in with the formal taxonomy is that based on anatomy alone, those differentiations between species in the same genus that a taxonomist would say, yeah, they're different species, seems to be about what is being determined by the mitochondrial DNA in terms of being 2 or 3, 4% difference. And so there's an alignment there between what the DNA barcodes are saying and what the formal taxonomists are saying is two species. So, yeah, you're not going to go to the point where you're not saying they're species because you haven't demonstrated whether they can reproduce or not. There's got to be a bit of pragmatism here. And but the thing that the DNA barcoding's got for it is that it's a constant once you've got it, whereas taxonomists are always arguing with each other about what the name is, and there's not enough of them, and they're getting older and they're not being reemployed. And so at a time when we need more taxonomy to determine all the biodiversity of the world so we can better protect it, taxonomy unfortunately is not getting the support that it really needs. And this DNA barcode approach is a way to get what we need quickly and comprehensively, and it's unequivocally. So it's a bit of a race against time to try, because we know that we're losing a lot of biodiversity. And so it is a bit of a race against time to try and get identifications for all the species on Earth. This is a big biology question. Some of the finest minds in biology have ruminated over this for decades as to how many species there are on Earth, So it's very profound and very important as well in terms of conservation, I think.
So I suppose if you actually got machines that can actually see differences in within a species, within you can therefore get a count of individuals. Is that it? Like you can say, well, here's individual 1, here's individual 2, here's individual. Is that how you can? The interesting thing is not only can you determine species with this, but you can also determine the numbers of a particular species. That's right, isn't it?
Well, that's what biodiversity is, isn't it? Really, it's the number of species that you have within a habitat.
Yeah, that's the biodiversity.
And that's biodiversity, and then they fulfill all those ecological processes that I mentioned beforehand. And we know that the more biodiversity that you have, the more resilient your ecosystem is if it's impacted upon. So it takes a hit. Some species disappear, but other species are still there, and they can still fulfill the ecological function, because there's a lot of ecological redundancy where you have a lot of biodiversity, and so it makes it makes habitats more resilient to stresses and strains. Yeah.
But this DNA analysis that we're getting can tell us how many bees you've actually counted, can't it, of particular species, or arachnid or something. Is that right? It can give you the volume?
Because the thing is that it's okay. We're taking a sample, okay, but every one of the individuals we collect is identified. So we're getting biomass as well. Biomass is as important as biodiversity.
Yeah, that's what I'm saying. Yeah, and getting biomass as well as
biomass, because ecosystem function is a function of biomass. The higher the biomass, the healthier the ecosystem function is. Excellent. And so that's why we're doing both. Now, the biomass is determined by AI, apparently, back at the University of Guelph. So from the samples that we send. And so for each packet we send after a week of sampling, there's about 600 to 800 individuals in that packet of that they then process individually. But they make an estimation of the biomass as well. And we know from just anecdotal observations that the biomass drops quite dramatically during the wintertime.
Yeah.
And so, that's pretty obvious. Now, the thing is, we don't have I don't believe we have a database in Australia to say, well, whether biomass is increasing or decreasing. But if you think about when you were a kid and you drove up the coast with your parents, all the insects that you used to get on the windscreen.
Right.
And now when you drive up, you get 3 or 4.
Yeah,
So
that's exactly right.
Yeah. So I think something is seriously happening with insect biodiversity on the coast, just from those sort of observations, unquantified observations.
Actually,
So,
Brian's put an interesting piece of information in the chat. Brian, would you like to tell us about the
silverfish
with the silverfish?
No, it's just a guy I ran into the other day who was absolutely totally crazy about, of all things, silverfish.
Right. Okay.
And there apparently are hundreds of them that are still to be described.
Yes, exactly. That's the thing. See,
they're very primitive too.
They are very
And
primitive.
even older than cockroaches.
Yeah. Yeah, that's true. Yeah, so there's a lot to be done. Look, we've tried to work with the Australian Museum in terms of DNA barcoding their collections, but they're not real keen. So I don't know why. I think a lot of traditional taxonomists feel a bit threatened about the DNA barcoding approach. That's the personal impression I have. I know that when Paul approached the CSIRO in Canberra about DNA barcoding, they were completely open to it. And so he's DNA barcoded a lot of their samples in Canberra. But the Australian Museum, not at this point in time.
Okay.
I mean, the door is always open, but not at this point in time. So if you see him again, say, "Oh, how would you like your silverfish DNA barcoded?"
He wasn't. He said you could get a lot of negative results. He told me, and I thought I won't chase this one down.
I don't know what that means. Yeah, look, I've never heard that. But
DNA barcoding has helped clarify species numbers more than the other way around. I mean, the classic taxonomists doing things.
Sure.
You've only got to think of your bird books you've had over the years, and the way the names of subspecies and even species change, it's DNA that has helped resolve some of those issues about whether, the eastern honeyeater is the same as the western or subspecies of it or whatever. It's going the other. It's not that the taxonomists will give us the answers. No. DNA stuff has told us the relationships between them all, and has helped solve all those tricky sort of questions.
Yes, and the economics of it all. It's $2 an individual sample, and the cost is declining. It's quite remarkable, the adoption of these new technologies coming out of, I think it's Oxford nuclear pore technologies are enhancing the throughput. And now you can buy a portable machine. So you can go up to, a billabong in the Northern Territory, and you can take a water sample and you can run it through. And within 24 to 48 hours, it'll tell you all the fish species that are present. So that's not DNA barcoding, but it's based on having a barcode library that you can refer to. And so you cannot deny the newer technologies that are coming around and solving a lot of these questions, getting answers to the questions that taxonomists have been arguing about in conferences for decades.
Right.
And we sort of don't have time to go on like this anymore. So anyway.
Just following on from what Gary said, DNA has created a whole evolution, a whole revolution actually in the botanic world. You go to the botanic gardens and analysis of plants and their genetic linkages going back it's all DNA now. And it's amazing.
Yes,
what about Odonata, the dragonflies? How do you think we can if you put traps or some different type of design, say down their water courses, might you get
look, I don't know. I don't know if we might have caught some, but I guess we weren't where we put our nets. They weren't really that close to water anyway, and so I don't know about that. I mean, I'm sure there's other things that could be sampled as well. using different approaches. I guess at this point where you want to run an international program using a well-defined protocol that's easy to deploy, and anybody in any sort of country of any state of development could still make it happen. I'm sure that's part of the deliberations as to why the MalaysNet was chosen. And that approach, because I know that Paul Hebert uses a lot of other techniques for collecting different taxonomic groups of insects as well. But for the International Barcode of Life, that's not what he decided to do. Yeah.
So it's not dropping a blanket underneath a tree and bashing the tree and catching what falls out.
Not exactly, no. Yeah.
Well, if just looking across the audience, it doesn't seem like there's another question.
Okay.
Or I think everyone would join in with me in first of all thanking you for a wonderful and very lucid and very clear and wonderful presentation, Ross. There's some wonderful slides there that I think will illuminate us for all time. And thanks also for your extensive coverage of the questions that we've provided you with. So it's been
it's a very stimulating to have the discussion with people about this. So I really appreciate it.
Gary's waving goodbye.
Thank you, one and all.
No, I think that was a clapping hand.
The one hand clapping.
The one hand clapping.
Thank you very much, Ross.
That was great,
fantastic.
It's a pleasure. And look, I mean, there's more results coming in all the time. And again, we're going to be getting nearly as many results coming in from the rainforest site. So watch this space, because I'm sure we're going to find a lot of other exciting things along the way as well. So cheers. Thanks very much for your attention.
Thank you for pioneering it too here, Ross. Thank you for getting underway.
It was a golden opportunity to get to pull in these resources to get something like this for the Royal. So anyway, thanks very much.
Okay.
For those of you who were on one of my walks before I had my knee operation, I think we walked past one of Ross's traps, didn't we? Yes. And Ross had escaped his trap before we came
that's right.
back.
Yeah. I remember. I saw Valerie. I think you'd walked ahead, Brian, and Valerie saw me, and I showed her the trap. On the little Marley track.
Everyone who started at the south end of Lady Carrington has seen Ross's traps. In fact, I've been questioned about that and gone up and photographed it and all the rest, and certified it wasn't a vagrant living there or a
Yeah, that was the one Melissa was sampling, and she usually picked up a leech as well for her efforts. So.
Oh, good. Did that get barcoded?
No, we didn't do that. It's not. It's outside the protocol. So.
It doesn't have enough legs.
Just an insect joke. Just an insect joke before you all go.
No.
That's it.
What did the earwig say as he went over the cliff? Here we go.
Oh, very nice. Yes,
that's it. Good
Thanks
night.
for that.
No more.
Okay.
Thanks,
See you later, Ross.
Okay, cheers. Bye-bye.
Okay, good night. Recording stop now.